Welcome to the Sand Science Experience, where we explore the latest and most exciting papers and preprints in science. And hey, right off the bat, if you find this deep dive insightful, please do hit that like button and subscribe. It truly makes a difference, helping us bring you more fascinating discoveries like this one. Today, we're diving headfirst into a topic that honestly sounds a bit like science fiction: small heat shock proteins, or SHSPs. Could these tiny cellular defenders hold the key to, well, a magic bullet against aging and disease? It's a really tantalizing question, especially when you look at the potential in simpler organisms. But here's the twist, and it's a big one. The story in humans presents a surprising and frankly critical paradox. It kind of turns our initial hopes upside down. So our mission today is to unpack this intriguing puzzle. We'll look at the incredible promise in model organisms and then tackle the complex, sometimes contradictory picture in us humans. You're basically about to get a shortcut to being truly well-informed on this cutting-edge area of biology. Okay, so to really get what we're talking about, let's start basic. Imagine your cells are like these bustling little factories. Inside, there's just constant activity, making proteins. They're the machinery, the building blocks, pretty much everything. But like any factory line, things can go wrong. Proteins get damaged, misfolded, they stop working right, especially under stress, like a power surge or bad materials. That's where molecular chaperones step in. Think of them as the quality control team. Super important. They make sure proteins fold correctly, or if they're beyond repair, they help get rid of them efficiently.
Exactly. And what's really fascinating within that team is this special group, small heat shock proteins, SHSPs. These aren't just run-of-the-mill chaperones. They're highly adaptable. They respond rapidly to cellular stress. And for our deep dive today, we're zooming in on one specific SHSP, Drosophila HSP22. Now, this particular protein has a very specific address. It lives exclusively in the mitochondrial matrix of the fruit fly.
Okay, the mitochondrial matrix. And why is that location right there in the cell's powerhouse? Why is that so important?
Well, think about it. Mitochondria are the energy generators, right? They make ATP, powering almost everything, but doing that creates stress. They produce reactive oxidant species, ROS, which are kind of like cellular exhaust fumes. Very damaging.
Ah, okay. So they make power, but also make damaging byproducts.
Precisely. So having HSP22 inside the mitochondria makes it absolutely critical for keeping that vital organelle healthy, especially as the organism gets older. It's like having a dedicated repair crew right inside the engine room, preventing damage from the inside. It's crucial for maintaining what we call proteostasis.
Proteostasis. That's keeping the protein balance right.
Yeah, exactly. The whole balance of making, folding, functioning, and getting rid of proteins within the mitochondria. HSP22 is key to that.
Right. Now here's where it gets, well, really exciting for anyone interested in longevity. The fruit fly stuff. Drosophila melanogaster. Some truly eye-opening experiments there.
Oh, absolutely. The evidence is incredibly robust. When you overexpress HSP22 in flies, meaning you tweak them genetically to make more of it, their average lifespan doesn't just nudge up, it jumps significantly. We're talking around 32% longer life.
30%, that's huge.
It is. And it's not just about living longer, it's about a longer health span. These flies stay active, their locomotor activity holds up much longer into old age. Plus, they show better resistance to various stresses, including that oxidative stress we mentioned.
So more HSP22 literally means a longer, healthier life for a fly. Simple as that, it seems. But what about the flip side? What if you remove it? Take HSP22 away completely.
Good question. And yeah, the contrast is stark. If you eliminate HSP22, what we call a knockout fly, the effects are dramatically bad. These flies suffer a striking 40% reduction in lifespan, a huge cut.
40% shorter. Wow.
Yeah. And beyond just dying sooner, they show faster functional decline. They're much more sensitive to stress. It really highlights how essential HSP22 is for them.
Okay, so it's clearly vital. Are these effects general, or are they linked to specific tissues, or maybe when the protein is active?
Both, actually. Really interesting studies show that if you target HSP22 overexpression just in the motor neurons, the nerve cells controlling movement, that alone is enough. You still get about a 30% lifespan extension and better mobility.
Just in the motor neurons, that really points to neuronal health being key for aging overall, doesn't it?
It certainly does. And the timing. Absolutely pivotal. HSP22 needs to be activated early in the fly's adult life, before about day 4.
Day 4, that's really early.
It is. If you try to boost it later on, the lifespan benefit is much, much smaller, almost negligible. It strongly suggests that you need to get in there early, prevent the damage before it really accumulates. Preemption is key.
So how is HSP22 actually doing all this? What's the cellular mechanism? What's the magic?
Right, the how. It seems to act on several crucial fronts inside the cell, particularly the mitochondria. First, it champions mitochondrial proteostasis. It's a chaperone, so it helps mitochondrial proteins fold correctly, prevents them clumping up into damaging aggregates, keeps the machinery running smoothly.
Okay, prevents protein gunk. Makes sense.
Second, it boosts stress resistance. It ramps up the cell's own antioxidant defenses, boosting those harmful ROS, minimizing oxidative damage. Less rust on the machinery.
Got it. Better defenses.
Third, it activates something called the mitochondrial unfolded protein response, or UPRMT.
UPRMT sounds like an alarm system.
That's a great way to put it. It's the mitochondrion's internal alarm for protein stress. When things get messy, UPRMT kicks in, triggers a rescue effort, more chaperones, refolding, getting rid of bad proteins, prevents a meltdown.
Okay, an internal rescue squad. And you mentioned a fourth thing.
Yes, finally, it's involved in autophagy and mitophagy. These in the cell's recycling programs. Autophagy cleans up general cellular junk, while mitophagy specifically targets and removes faulty mitochondria. HSP22 helps these processes run efficiently, keeps the whole factory clean.
Wow, okay, so it's doing a lot, maintaining proteins, fighting stress, running quality control, and taking out the trash. No wonder it extends life in flies. Now, that fly data is super compelling, but is it just a fly thing, or do we see hints of this in other simple models, like, say, the nematode worm C. elegans?
Yeah, we do see similar principles, largely conserved. Now, C. elegans doesn't have one single exact mitochondrial equivalent of HSP22, but it has its own family of SHSPs, things like HSP16.2, HSP12.6, that perform very similar protective jobs. HSP16.2 is often considered the functional counterpart, the functional ortholog.
Functional ortholog, okay.
Yeah.
A similar job, different protein name. And do they affect worm lifespan?
They do. You see a consistent pattern. If you overexpress certain worm SHSPs, like HSP12.6, you can get lifespan extension. And if you reduce them, lifespan tends to shorten, matches the fly story.
Okay, consistency across invertebrates.
So it's more, if you boost HSF1, that's heat shock factor 1, the master switch in worms that turns on many SHSPs during stress, that also extends lifespan. So it really reinforces this idea that using these chaperones for longevity is a deeply conserved ancient biological strategy.
Right. Flies, worms. The evidence seems to be building a really strong case for these proteins as anti-aging powerhouses. Almost, too good to be true. Which brings us to the big question. What about us, humans? Is there a clear path to translate this? Does the magic bullet work for us?
Well, this is exactly where the narrative shifts. It becomes much more complex and, honestly, quite challenging. In humans, the protein HSPB8 is generally seen as the closest functional relative to the fly's HSP22. But, and it's a big but, our SHSP family is larger, at least 10 members, and way more complex. Each one might have slightly different roles, different places it works.
More complex. Okay, so maybe not such a simple story.
Exactly. And that complexity might be why a simple more is better idea doesn't quite hold up.
So do studies actually show that people with naturally higher levels of HSPB8 or other SHSPs live longer, like the flies?
Well, no. Not directly, and this is the core of the human puzzle. If you look at massive human genetic studies, GWS, genome-wide association studies, scanning thousands of people, they just haven't found strong evidence linking common variations in our HSPB genes to human longevity or healthy aging in the general population.
No link. So, unlike flies and worms, having common genetic tweaks in these genes doesn't seem to make a big difference to how long most people live.
That appears to be the case, yeah. It's a major divergence from the invertebrate models.
Wow. Okay, my brain is trying to square that circle. It feels like a complete reversal.
Really?
But you mentioned disease earlier. Is that where these proteins become relevant in humans?
Precisely. This is the critical and really surprising turn. Instead of promoting longevity, rare mutations in human HSPB genes, specifically HSPB8, but also others like HSPB5 and HSPB1, are directly linked to severe, often early onset neuromuscular diseases.
Diseases, not longer life. What kind of diseases?
Things like debilitating myopathies, muscle diseases, neuropathies, nerve damage, even cataracts. So a pathway that clearly promotes long life in simpler animals can, when disrupted by specific mutations in humans, cause devastating illness.
Okay, hold on. The same family of proteins that make flies live longer can cause crippling diseases in humans if they're mutated. That's more than a paradox. It's almost biologically unfair.
It is a profound paradox, isn't it? And it immediately flags a huge potential safety issue for any therapy ideas. These disease-causing mutations often create what's called a toxic gain of function.
Toxic gain of function, meaning the mutated protein doesn't just stop working, it does something actively harmful.
Exactly. Instead of just being absent, the mutant HSPB proteins often misfold and start clumping together, forming aggregates. These aggregates basically overwhelm the cell's quality control systems. Imagine the quality control crew suddenly turning into toxic sludge that jams up the whole factory.
Okay, that's a scary thought.
It is. And it strongly suggests that just blindly boosting SHSP levels everywhere in the human body could be really detrimental. You might push the system into a diseased state, not a healthier one. And remember, HSPB8 is naturally found in high amounts in tissues like muscle, heart, and neurons. The very tissues hit hardest by these disease mutations.
Right. So the idea of a simple magic bullet gets really complicated, even dangerous, fast. The potential is there based on the flies, but the human reality is risky. Given this whole complex picture, what are the potential ways forward? Are people still trying to harness these proteins carefully? And what are the biggest hurdles?
Absolutely. The potential is too intriguing to ignore, but the approach has to be incredibly nuanced. Pharmacologically, one idea is to indirectly nudge the system. Instead of adding more SHSP protein, you activate the master controller. So compounds that activate heat shock factor 1, HSF1, the main switch for the whole heat shock response are being looked at.
So you trigger the body's own natural stress response, which then makes a balanced set of chaperones.
Exactly.
Yeah.
It's potentially a more subtle, regulated approach. Also, things like HDAC inhibitors. Trecostatin A is one example, have been shown to boost HSP22 and lifespan in flies, so that's another angle. Leverage existing systems.
Okay, indirect activation. What about more direct approaches, like gene therapy? You mentioned that's possible now.
Gene therapy is definitely on the table, yes. There have been mouse studies, for instance, using AAV vector-safe viruses used for delivery to get HSPB8 into specific tissues, like delivering it to the hippocampus in the brain improves cognitive function in mouse models of diabetes.
Yeah.
So tissue-specific delivery, technically feasible.
What's a dosage, right? And getting it only where you want it.
Precisely. That's the massive challenge, especially given the human paradox. You need exquisite control over dosage and tissue targeting. Too much, wrong place, wrong time, could potentially cause harm. The therapeutic window seems very narrow.
Which brings us back to safety. What are the absolute biggest red flags if someone did try to systemically boost these proteins in humans?
Based on what we know, there are two major concerns that keep researchers cautious. First is a potential cancer risk. SHSPs help cells survive stress, which is good for normal cells. But that same survival advantage can help cancer cells evade programmed cell death, apoptosis. Some studies have linked SHSP overexpression to increased malignancy, helping the bad guy survive.
Okay, cancer risk, that's serious. What's the second one?
The second is the direct risk of neuropathy and myopathy. Given that human mutations cause these exact problems, there's a real concern that artificially increasing SHSP activity, even if it's the normal protein, could inadvertently trigger similar nerve or muscle damage if it's not perfectly regulated, mimicking the disease state.
So a very fine line between potentially helping and potentially harming.
A very fine line. It demands extraordinary preclinical safety testing, much more than usual. So wrapping this up, what's the verdict on the magic bullet idea for SHSPs against aging? Well, the core mechanism, stress resistance. Proteostasis are fundamental and conserved. That makes them super attractive targets. The fly and worm data is compelling. But the human data throws up a huge caution sign. Direct systemic boosting looks risky because of those clear links to severe human diseases when things go wrong. It's a powerful pathway, but messing with it directly in humans needs extreme care.
It really leaves us with a fascinating thought, doesn't it? Instead of this blunt, more is better approach, which seems problematic in humans, perhaps the real magic isn't a magic bullet at all. Maybe it's in understanding the subtle, natural ways these proteins are regulated in healthy, long-lived individuals. Or how things we already know are good for us, like exercise, maybe sauna use, certain diets, how they gently induce these protective pathways, beneficial stressors. Could we design therapies that mimic those gentle nudges rather than using a sledgehammer? Maybe focusing on compounds that mimic exercise effects or nutritional tweaks. That might be a safer, more biomimetic way to tap into their anti-aging potential without accidentally triggering disease. It makes you wonder, doesn't it? What subtle strategies might you think of to gently nudge your own cellular stress responses rather than trying to force them? Something to ponder. Anyway, if you enjoyed this deep dive, please remember to like the video and subscribe. It really helps support the Sand Science experience and lets us keep bringing you these explorations. Thanks so much for listening, and we'll see you in the next deep dive here on the Sand Science experience.